Next, would you please welcome or join me in welcoming Dr. Vinod Prasad from Bluebird Bio for an update on gene therapy for cerebral ALD.
Okay, well, good morning, everybody. Hope you're having a good morning so far. I want to thank my friend Dr. Tim Olson for a wonderful talk and also for wearing a tie. I was feeling a little bit out of place this morning when I walked in with a tie on. So thank you to Elisa and to Miranda and the whole of the ALD Alliance team for inviting us to speak about gene therapy. It's really important that we are as transparent and as clear in providing information. I also want to thank you for putting this wonderful agenda, which clearly has depth and breadth of various topics that are interested to the community. And so it truly is a standard of care conference, as you're calling it. And for bringing all these experts to the table and having an open and clear communication with the patient and families, and make it really one of the most important events in the ALD calendar. So thank you very much for doing that. I feel like I'm amongst friends. This is my first ALD Alliance meeting. I came yesterday a little bit late, but I walked in. I certainly know a number of people. I walked in. I got some silent nods of hello. Then I also got virtual hugs. I got a couple of real hugs. And so I felt like I'm with friends. My only disappointment was that I did not get any flying kisses. So I got to work harder on making this connection. So the slide is up. I've been taking care of, for folks who don't know me, I've been working with Bluebird Bio now for the last 8 months. I've been doing transplants for more than 20 years, and I've taken care of a lot of patients with adrenoleukodystrophy, other metabolic diseases, as well as leukemia and MDS and a number of other diseases over many years. So I've taken care of a lot of children with MLD, and it's really, I think, been an honor and a privilege to be a part of this community. And I really thank you for being so committed, all the different stakeholders, in making the lives of our patients better. As I said, during these many years of taking care of patients with ALD, I've had two heartbreaks, two categories of heartbreaks. The first one is when you have a patient who comes to you with whether it's adrenoleukodystrophy or a similar disease, when you know the transplant is going to be helpful, but you can't really do the transplant because either the disease is way too progressed and you think that the risks are really, really high of doing the transplant, the risk of mortality related to transplant, and you cannot do transplant, or even if you do transplant, the outcomes are not going to be that great. And that heartbreak is really something I have felt, many of the families have felt. I was fortunate to take care of Aiden Seeger, Elisa's son. I was part of the treatment team at Duke that took care of him. He was one of the most handsome boys that I remember, with a really kind face, and I can still feel his hug that he gave me the first time I met him. That left a lasting impression. But imagine what impression it must have left his life on his mother, who took it on herself to do everything to bring newborn screening to all children born in the United States so that patients who need transplant or other therapies can actually get them at the right time. And so thank you, Elisa, for being the driving force and the guiding light in newborn screening for adrenoleukodystrophy. And thank many of you in the room who really have been working to make it a reality. And now, big round of applause to having did I hear 44 states now? 42 states out of 50. That's a great accomplishment, so thank you for doing that. The second heartbreak that I've had is you do the transplant, but because of complications, there is transplant-related mortality that Dr. Olson talked about. There's risk of graft failure, that you do the transplant in a technical sense is successful, but because of toxicity, the patient doesn't survive. And that is what needs to be addressed. And I think from what we heard, gene therapy may allow less toxicity. But I've also learned a lot of things as I have moved from, although I have a dual role, I've moved to the biotech industry. And I'll come to that in a second. I'll take a sip of my tea while you read my disclosures. So I'm a full-time employee of Bluebird Bio, and I'm speaking on behalf of Bluebird Bio right now, and I will not be giving any medical advice. And so one of the things that I've learned in biotech is that it's a very well-regulated, for good reason, well-regulated industry where we have a lot of limitations. And one of those limitations, or I actually should go through all of these limitations, and that is that regulations prohibit the pharma companies from promoting a product in a manner that is not consistent with the prescribing information, which was approved by FDA. So I'll talk a little bit more about that, and I think this is really important. Also, FDA approved Skysona under accelerated approval. And the population of patients considered on label was much smaller than the total experience that we have with Skysona. So when I talk about efficacy, I can't really talk about the whole population. Dr. Olson, Dr. Duncan can talk about it, but I cannot. I can only talk about what is on the label, and I'll mention that a little bit later. Because it's an accelerated approval, we have a responsibility to continue to provide updates to FDA on a regular basis, and we are doing it. And also in that regard, it's really important that we follow the patients on a long-term basis, and I will talk a little bit about that too. The best source of information, I think, is your healthcare team, and we do communicate with the community through scientific presentations, which is really important for the healthcare practitioners, and they have the ability to look at all of that data. And we also try to go to the community meetings and talk about gene therapy. So in that effort for transparent communication, I point out 3 different presentations that we have had. The one in the middle is the ALD Connect conference, where I presented last year in November at Houston, and I saw many of you there in that meeting. In addition, Dr. Florian Eichler, who is in the room and a driving force in this field on the neurology side, he presented the efficacy and other data at the CNS meeting in September of 2023 in Vancouver last year, and that abstract is also available in the public domain for your healthcare providers, and they can look at it and then convey those information to you, to the patients and the family very clearly. And just last month, Dr. Duncan, who has been leading the transplant and the gene therapy in many ways for adrenoleukodystrophy, presented the safety and also data related to mobilization and apheresis for gene therapy on all of the patients, 67 patients, at the tandem meeting, which is a transplanters meeting. So what we are trying to do is to communicate with the community, as well as communicate with the neurologist, as well as the transplant doctors, the 3, I think, pillars of treatment teams. So I was asked to give an update on the gene therapy. Whenever any drug is approved by, can you see my pointer? Yeah, okay. Whenever any gene therapy, any therapy is approved, the FDA produces a label. It's called USPI. And that label is a way to communicate all the information about the drug, whether it's gene therapy or any other drug, to the patients, to the healthcare provider. And it's a document that is really held high when we think about in a biotech or pharmaceutical company as a way of communication with the family. So it has to be accurate and it has to be very focused. In that, you will notice that Skysona is indicated to slow the progression of neurological dysfunction in boys 4 to 17 years of age with early active cerebral adrenoleukodystrophy, and then we go on to describe what an early active ALD is. There are also limitations of use where, on the basis of the data that was reviewed by FDA, FDA determined that there will be some limitations, that it may not work for everybody, and those limitations are also listed in the limitation of use. And if during that discussion with FDA, FDA determines that there are some significant risks associated with a particular drug, that goes into a boxed warning, which is on the top page of the USPI or the label. And for Skysona, there is a box warning for hematological malignancy or blood cancer. To develop a gene therapy program, which really is cutting edge, it's new in the class, it's never been done before, it takes a long time. It took many years of the lab team at Bluebird Bio and the scientists to develop the framework of how to do the gene therapy. And then the first set of studies, which are listed on the top row, are the studies to look at the outcomes of patients who were either untreated or were treated with allogenic transplant, which is considered standard of care if there is a donor available. Subsequently, there were two studies which were done, as Dr. Olson mentioned, ALD 102 and ALD 104 studies, with some difference between them. And as was required by FDA, these patients, all patients who were on 102 and 104, then rolled on to a long-term follow-up study called LTF 304. So they can be followed for another 13 years. So there is a total of follow-up of 15 years for all of these patients. In addition, because of our commitment to long-term follow-up of these patients, ALD or REG 502 is a protocol that was opened to follow all patients who have received commercially approved Skysona product. So just to underline that there is a need for continued follow-up and there are plans for continued follow-up. Before I go into gene therapy, I think it's important and to some extent redundant now because of what we heard from Dr. Olson, that allogenic transplant is, while effective in certain situations, also has toxicity associated with it. But I will just highlight the important point here on the first slide or the first picture here, that MFD-free survival is better if you do transplant early. The outcomes in terms of MFD survival as well as overall survival are much lower if transplant is done later in the course of the disease. So if the LES score is more than 9, the transplant outcomes are not going to be that good. In addition, in that paper which was published after the ALD 103 study, we saw that there was significant risk of acute and chronic graft versus host disease, as well as transplant-related mortality and graft failure. I've already mentioned the 102 and 104 studies and the long-term 304. The important point I want to highlight here is that these patients have been followed for a long time. So we have almost 10 years of data, the longest patient having follow-up of almost 10 years. Patients receive conditioning, as we just heard. Prior to that, cells are collected. Those cells are then transduced, and then after conditioning, those cells are infused into the patient, and then those cells will engraft and develop hopefully microglial cells that are gene modified and will go to the brain and then correct the defect that needs to be corrected. One of the reasons why we wanted to do this presentation is to give you an update in the effort to be as transparent as possible from Bluebird Bio to the ALD community. And so some of the data that I will show you is from Tandem meeting. Actually, before that, I will show you some data that was part of the USPI. When you look at this MFD-free survival graph, the one which is dark line tells you the MFD-free survival with Skysona, while the dotted line shows you those patients who did not get any treatment or untreated patients. And then this one shows you the outcomes of Skysona treated patients with a dark black line, and then compared it with the allotransplant from matched versus the mismatched donor, and the mismatched donor outcomes were lower than those with the matched as well as with the Skysona. I don't think, in the interest of time, I need to go into a lot of details, but these slides will be available to you if you want to have dig deeper into this and want to look at this in any detail. This session, I think, is being recorded, and all these slides will be available to you. I think Dr. Alston already mentioned some of the differences between 102 and 104. I would skip this other than to say that there is, in terms of the patient, there wasn't that much of a difference. The median age in 102 was 6 years, and in 104 it was 7 years. Not that much of a difference. In the less score, the median was 2 in both studies, and the range was 1 to 9 in one and 1 to 7.5. Another one, we already saw these slides. Essentially, there are 3 points I want to make. One is, in terms of engraftment, there wasn't that much of a difference between 102 and 104. Number 2 is that all patients engrafted for neutrophils as well as platelets between these two protocols, and that all patients were discharged from the hospital after having engrafted. Whenever you give chemotherapy, whether it's for transplant or for gene therapy, there are a number of toxicities, and we heard that there were some more toxicities in 102 in relation to chemotherapy-induced GI toxicity or gut toxicity or blood toxicity. There were also some patients who had delayed platelet engraftment, which means that 14% of the patient had a platelet count of less than 50,000 beyond 60 days after treatment with Skysona, but then subsequently improved. I want to spend some time on hematological malignancy. Hematological malignancy is a serious diagnosis. It's scary. And in simple words, it's blood cancer. There is a box warning for hematologic malignancy with Skysona, and we have talked about this very clearly. Hematological malignancy, including life-threatening cases of myelodysplastic syndrome, has occurred in patients with Skysona. As Dr. Alston's slide showed, the data was presented in some detail on 5 patients by Dr. Duncan's presentation at the Tandem meeting. Those are available to all of you. In addition, there is an additional case of hematologic malignancy with acute myeloid leukemia that was reported 3 weeks ago, which is being studied very carefully for possible role of lentivirus integration. It's really important that we monitor the patients very closely post gene therapy, whether it is for on the trial or for the commercial patients, and also monitor them with a number of different parameters in terms of integration of the gene into the blood and bone marrow and so on. And there is well-defined protocol for this, and it is available. I do want to mention these 5 cases. At the time of transplant, they range between 5 to 13 years of age. At the time of diagnosis of hematologic malignancy, they range between 11 to 15 years. All of the patients were subsequently treated with either chemotherapy initially, followed by allotransplant, or with allotransplant directly, and that depends on what is the condition of bone marrow at that time, and that was determined by their physicians. One patient passed away because of complications related to graft versus host disease after MDS had been resolved. Other patients also, the MDS is resolved, and as of last follow-up, they are alive and doing well. So, in the light of hematological malignancy, what are the things that we have been doing at Bluebird Bio? We have had continuous commitment to share the data with the Data Monitoring Committee, which is charged with study oversight, and we have been providing them updates. We have informed the investigators, treating physicians, we have informed the investigators, treating physicians at QTC as well as the regulatory authorities, including FDA, about this. And the physicians then have informed the patients based on their institutional guidelines. We have updated the CALD community, and we do provide this information to FDA on the regular basis. We are investigating into the cases to determine the role of treatment, including the latest case I just mentioned. We are exploring other factors that may increase the risk of malignancy, and we are sharing ongoing updates from the investigation with the clinicians, and then clinicians also are communicating with patients as we make determinations about all of the patients who have been treated and the patients who are going to be treated. It's really important to monitor for hematological malignancy, and all of this information is clearly communicated and available. So I do want to come to this in the perspective of what we know and where we stand. Gene therapy for ALD is an important treatment. We have seen that it's been effective. It's been approved by FDA for slowing neurological progression in patients aged 4 to 17 years of age. It has some toxicity, hematological malignancy. Allogenic transplant is also effective, and it has its own complications. There are experimental therapies that are being developed. Between these two therapies, it is really important that the physicians and the families have all the information, as we have just shared, and make a decision about what is best for your child at any given moment. And we will continue to provide any updates. So as I said, in the benefit and risk assessment, Skysona is indicated to slow the progression of neurological dysfunction in boys 4 to 17 years of age with early active cerebral disease. And this indication was approved on the basis of efficacy as measured by 24 months major functional disability free survival. We need to continue to monitor these patients, and as a part of that, there is the new study that is just open. I think Boston Children was the first site, and the intent is to continue to open it at all sites where patients are being treated. So we have a close follow-up, and we can learn and try to mitigate the risks as we move forward. With that, I want to thank all of you. I want to thank the patients and the families who have participated in these studies and continue to be our partner. And what happened here? Okay. And I also want to thank Bluebird Bio family, who have really, in last 20 years, have worked hard at the science level to develop this therapy, with the clinical research of bringing all of this through FDA approval. I can tell you, just being there for 8 months, that everybody at Bluebird Bio really is focused on what is best for the patient. Their heart bleeds for you, and it beats for you. Thank you very much. Any questions? Oh, yeah.
Thank you very much for that talk. The cost. What does the landscape look like with regards to attaining this therapy, given the various economic situations we have across the country with patients?
Sure.
You can comment on that.
I can certainly talk about it briefly. There are experts at Bluebird Bio who really work with the payers and getting the approval through either a third-party insurance or through government insurance. But so far, my understanding is that those patients who require treatment have been able to get it approved through their third parties. And I think Dr. Olson, who is taking care of some patients, and Dr. Duncan, who is taking care of some patients, they would be able to speak more to this.
We have a question on Zoom. Dr. Waldman is saying, wonderful talk. Can they share how many boys have been treated with Skysonis since the approval?
I don't have that information, unfortunately. I think, not a question for me, I'm sorry, I just don't know. But I think maybe Dr. Olson, Dr. Duncan, who have treated, it's just small numbers. Certainly, given the rarity of the disease, I wouldn't expect it to be a large number. But I don't think that a patient has been denied treatment. If somebody came to a qualified treatment center who needed a gene therapy, they have been able to get it. Is that a fair way? Yeah. Okay.
I was just curious about talking about the cutoff for receiving treatment. So in the setting of allotransplant, it's always been discussed the 9 as sort of being a cutoff, but I kind of look at it as a soft cutoff because I know so many amazing ALD boys who presented with less scores higher than 9, but the medical team, after evaluating their clinical presentation, decided that there was the possibility for a good outcome. For boys presenting with a score that might be slightly higher than a 9, if it's still determined there might be the possibility of a good outcome, is that going to be a hard no because of the way the FDA approval was structured? Or are there abilities to appeal based on a case where you might possibly think a good outcome might be obtainable for a child?
That's a very important question, right? That there are specific guidelines. What I can say is the decision for any particular patient of whether gene therapy or any treatment could be useful beyond what is on the label is a physician's decision and a family's decision. And I will leave it to the physicians and the families to make that determination. Okay, good. Well, thank you very much.